Earth actually comprise our most accessible example of a second type of a biosphere compared to our early Earth. And then finally, the culmination of this early evolutionary process was the first branch on the tree of life. So I mentioned the last universal common ancestor. That point in the tree, the root of the tree of life, just following it is the first divergence of a common ancestor into two different forms of life. And this branching process has continued for the last several billion years. And that's what's responsible for shaping our biosphere and allowing life to inhabit every possible environment on Earth. And so I contend that any extraterrestrial life form that would be detectable by any method that we have would have had to go through the same process that we see in early evolutionary history, or else it wouldn't have been capable of escaping and growing beyond the narrow environment in which it originated. And so that's all I have for an introduction about myself. And I guess, Heather, are you the next person in alphabetical order?
Heather, you're muted.
Yeah, I think I am. I was like, are we going by first or last name? So, because you would have been first in both cases. Yeah, thank you so much. I'm Heather Graham. I'm a research physical scientist at NASA Goddard Space Flight Center, and I'm an organic geochemist by training. My previous life was in paleobiology and paleoecology, and I now am in an astrochemistry laboratory because I'm really interested in understanding how to deconvolve organic signals from what we find native in space, just the kind of abiotic organic chemistry happening around us all the time. I lead up a group, the Goddard Agnostic Biosignatures Initiative. And that's if you've been attending this meeting, you know what that means. We're just trying to think about really basal ways of identifying life that aren't necessarily tied to the kinds of observations we have of life on Earth. So very different than the kind of work that Aaron is doing. And I didn't actually prepare any slides, and I feel very bad about that, but I do want to point out a couple of things that have come out of the group recently that we're really excited about as two agnostic metrics that we're interested in, and they're very computational as well and very chemistry based. One is a way of looking at molecules and gauging their complexity through as an intrinsic property of that molecule, and then expanding that out to assemblages or populations of molecules so that we can say how likely it was that that molecule or the assemblage would have originated from a living system. And we're also doing some work looking at how to employ scaling laws, which we think are very universal to life, and think about elemental stoichiometry in particles as being a way to understand if that particle is likely to have derived from life. That particle could be a cell or not. So that's a way of really, divorcing ourselves from the kinds of things that we might, just look at and try to think our life. Thank you.
Thanks, Heather. Over to Adrienne.
Hi, everyone. My name is Adrian Kish, and I am a researcher in Paris, France. I'm attending remotely at AbSciCon, and you can see here I'm at the Muséum National d'Histoire Naturelle, so that translates to the National Museum of Natural History in Paris. And I'm an extremophile microbiologist. So essentially what this means is that I'm looking for life in the weirdest, what we consider the most inhospitable places on Earth from a human point of view, and try to understand how it is that we have microorganisms that live there. And specifically what I look at is occasionally I get out to the field and actually get to collect samples, but most of the time I am in the lab and learning exactly the molecular machinery of what lets them live in this environment that a human body could not simply support. So what we're doing is we're looking really at a really detailed level through the machinery of life at a very basic level, and then trying to imagine now, is this something similar that could have arisen on another planet? What does this tell us about our own planet and the potential for life someplace else? So a couple of main points to keep in mind when we consider extreme microorganisms. The word extreme just means from our point of view as human beings. So we are still back in microbiology with the terminology is almost back at the Galileo level of, well, does the earth rotate around the sun or the sun around the earth? We're still very human-centric. We think everything revolves around us, and the terminology seems to reflect that. So what you have to remember is that these environments aren't extreme for the microorganisms that live there. They love this. Boiling acid, bring it. They love these environments. It's only us that doesn't like it. So for us, it's extreme. For these microorganisms, they are in exactly the environment they should be in. So astrobiology, what it lets us do is get out of our biases. It takes the human out of the equation and lets us look at life at a very fundamental way and takes away a lot of the things that we presume normally about life. We only know we have life on this planet. It's the only place we know that there's life, and there's life absolutely everywhere on this planet. So by looking at astrobiology, what that helps us do is to appreciate the rare and precious nature of life, and to appreciate the biodiversity. So a couple of things I'm really excited about now in my field. It's a great time to be a microbiologist because about every 6 months when I'm designing my classes for graduate students in microbiology, I have to completely redo our slides of what we know of as the diversity of life. It's amazing. We are developing new techniques that allow us to understand the diversity of life on Earth. So even our own planet, we're still rediscovering. And that's because we're developing new techniques that are much more sensitive. We can get down to the micrometer level and the nanometer level to understand machinery inside these microorganisms and how they can survive. So it helps us understand life on our planet and apply that to the potential for life someplace else. One thing that we are excited about in my lab is that soon we'll be participating with the ExoCube experiment on the International Space Station. That's going to be set for launch in the late 2023 or early 2024, depending on Crew Dragon's launch schedule. And so what we're going to be looking at is not only can microorganisms, living microorganisms, survive the radiation in space, and we're going to be using in situ monitoring on the space station to monitor them while they're exposed to space radiation. And that's going to be the first time we're going to be able to do that. And we'll also compare that to their biosignatures by using just molecules from their cells, from dead cells, and seeing how those react to the radiation in space. Again, being able to monitor that directly on the space station. And very excited eventually about ExoMars. Unfortunately, with the war in Ukraine, that launch has been canceled. We're excited to see that launch eventually at the next possible opportunity for the first time to look at, was there ever life on Mars? So those are a few things that I'm excited about now. And just to finish my introduction, there's just a quote from T.S. Eliot that I just think is so encapsulates astrobiology, which is a classic. We shall not cease from our exploring. And the end of all of our exploring will be to arrive where we started and to know this place for the first time. And that's what we're doing. We're looking out to understand our own planet and the life that's here.
Love it. Thank you, Adrienne. And our final speaker has heroically made it here from her opening plenary. Betül, go take it away.
Thank you so much. So I'm a biologist. I am interested in life's earliest innovations. Life is actually quite innovative, and it found really creative ways to make use of what is in its surroundings. We don't quite understand how, especially a difficult problem if you think about the long time scales. My lab creates experimental tools to resurrect ancient DNA to understand the first innovations of life. It goes back to thinking about the past of our planet as an altogether alien environment. As my colleague likes to call it, Sanjoy-san, that our planet's Earth's past is the best exoplanet. It's a very strange place. We like to study it, we like to understand it, but what is lacking is to quite grasp the innovation at the molecular level. So we do multiple things. As I said, we like to think of ourselves as molecular time travelers. We resurrect ancient DNA inside modern organisms to reprogram modern bugs to behave like they did once in the past. So we really want to understand this dance between life itself and the environment. I like to think of being a biologist on this planet as one of the most unique jobs there can be. This is the only place where you can study life in the entire universe. Astrobiologists are working hard to change that. And that makes our job and task a really very special one.
Thank you to our panelists. We're going to open the table to questions now from audience, and I see everyone turning on their camera. That's wonderful. If you would like to ask a question, you can raise your hand in Zoom or wave at us or just open your mic and jump in. So anyone ready to go? Alec.
Hi, I'm Alec Jonath from The Economist. Thanks for doing this call for us. It's
We
really
don't hear
interesting.
you, Alec.
Oh, I'm sorry. Can you hear me now?
Sorry, it's just me. Go ahead.
Yeah, so sorry, it's Alec Jonath from The Economist. My apologies. Thanks for doing this call. I really appreciate you telling us about your work. I'd love some examples because I think this idea of life as we don't know it is fascinating, because all we do talk about normally is life as we know it. So I wonder if, perhaps, Aaron, you might talk about some of the reasons why, when you said that life branches or should branch, why might it not? What molecular mechanism might have evolved so that it doesn't do that? So therefore we could look for that. And is that really life in that case? And I wonder with Dr. Graham as well, I wonder if when you talk about the complexity of molecules, looking for patterns, can you give me an example of what's a pattern of life and what's a pattern of just chemistry in the atmosphere, you know? So that I'd be really interested in some of that. And I've got more questions, but I'll leave it there for now.
Sure. Should I go first, or Heather, do you want to go first? Okay. So to answer your question of what might keep life from branching and diverging, and what could life as we don't know it actually look like at the same time, I mentioned in my introduction, and these are both great questions, by the way. I mentioned in my introduction that one of the areas we're going into is designing artificial life simulations to get at evolutionary principles that would have been really important in the early evolutionary history of life, but could apply to life now, life on other planets, our more sort of general perspective on life. And one of our studies using this model that was published a couple of years ago was looking at why life evolved its cellular organization in the first place. And we found that under certain selection pressures related to the sort of richness of the environment, that you would have had not only the lack of an evolution of cellularity, but a selection pressure, an evolutionary selection pressure against cellularity. That if the environment was rich in the kinds of things that the organism could directly use to grow and reproduce, then it benefited the organisms to be open to the environment. So the last thing you would want to do is be cellular. We take the cellular organization of life for granted today, but as I mentioned before, that's one of those things that actually evolved and under certain scenarios would have been adaptively beneficial. So to answer the second half of your question, what kept life from diverging into different lineages probably had to do with the fact that organisms may have been randomly swapping genetic material at too high of a rate to get the sort of signal of vertical inheritance, the sort of consistent lineage of genomic information that's required to pass on, to inherit material from a parent and then pass it on to an offspring. The random transfer of genetic material between living organisms can disrupt that process and could have led to a lot of evolution taking place before we could detect a divergence from the last universal common ancestor to the next most recent ancestors on the tree of life. Did that answer your question?
Certainly, thank you.
Thanks.
Betül, do you want to follow up, or I see Heather also wants to jump in?
I was just gonna try and riff off of Aaron's answer to address the question that you directed towards me as well. That when we're thinking about patterns of molecules, whether it be in their intrinsic molecular structure or how they are expressed in assemblages, is both of those things are really stemming from the same things that Aaron's talking about, selection mechanisms. We have these particular patterns that arise in organisms because there's been a selection mechanism towards the production of that molecule. And so a couple of things have to happen then. There has to be a way for an organism to jump an energetic barrier to even make that molecule. There should probably be a reason for it to be there, and it has to be unique depending on its job. There's a couple of jobs this isn't true for, but for most cellular jobs, it has to be unique from all of the other molecules in its environment so that it can do its job in the metabolism. So it can signal, so it can, be part of an information chain, things like that. So this idea of how molecules express patterns in biology different than in just physical systems really does stem from evolution. And so if you think about the kind of molecules that are being generated in space through abiotic process, or in the deep earth through abiotic processes, those are just following physics and assembling in ways that are easy through just kinetics to go through those energetic barriers. But life jumps those energetic barriers in order to make unique assemblages or more intricate versions of these molecules to make them different from the environment. And this is a way of thinking about not necessarily size or something like that, but how unique that molecule can be in its environment. So it's very much tied to evolution. And so that's kind of how I would relate this same idea back to chemistry.
I tend to think that I don't think we are harvesting the possible innovations and the history of life on Earth at the way that we can. Life samples DNA sequence space all the time. We are the outputs. What we see around us is billions of years of such sampling. And if you think about it, everything that we do today, and by we, I mean all biology, what we process, what we are able to do, relies on events that happened billions of years ago. And life's history there are certain singularities and evolutionary singularities that set the tone of life as we know it on this planet. That's the emergence of cellular life around 4 billion years ago. That is the oxygenic photosynthesis. It happened once. Life only found one single way to fix the inert nitrogen in the atmosphere. I think it is fascinating that there's a single enzyme that life relied on that even triggered the emergence of animals on this planet. And yes, that is going back to nitrogenase and nitrogen fixation. So there are certain singularities that happened in the history of life. And if you look at them, you will realize that there are gaps in between these innovations, in some cases, billions of years. I can count you maybe 5 or 6 of these major things, and that is it. But what we know is that evolution at the molecular level happens at the matter of seconds. Variation rather happens at the matter of seconds or minutes. So while we see these innovations that expresses themselves at the billions of years of time scale and very few of them, we know that what's underlying is happening at much faster time pace. We lack this overlap, the understanding of how this is even possible. So going back to life as we know it, I think it becomes a question from how to what. What possibilities can life do? What else can life do? Why did that life come up with a single way to fix all these atmospheric elements? There are no real answers to this. We don't quite know these fundamental questions and answers to this, but I think artificial biology and synthetic biology and evolutionary biology, such as experimental evolution, can offer insights into this. We need to understand whether these events were inevitable. We need to understand the degree of chance and determinism that impacted these events. We have to know what sort of planetary composition, what sort of inorganic composition and organic composition may give birth to these innovations, or whether they were restricted by the very planet. I think once we grasp that, we will be able to tackle a lot of problems head on.
Thank you.
Who would like to ask a question next?
I have a question, if I may. A lot of my friends like to play Wordle. I haven't done myself, but I just wondered if I could kind of adapt that game and ask you each if it's announced that an alien organism has been found. What is the first question? If you only had one question about that organism, what would that question be? I mean, to give an example, my first question would be, does it have a mitochondria? I think that's, kind of, to me, one of the most fundamental questions. But I just wondered if it might be enlightening to hear from you what question you would ask if you had just that one question.
I don't know. I feel like maybe
How much money do we got to answer this question, first of all?
I think you get 6 words if we're playing Wordle.
Yeah, that's the problem. I don't know anything about Wordle. I'm gonna do a terrible thing, Leo, and I'm not gonna answer your question, but I'm gonna tell you a little bit about something that we are using and developing as a community to try and tackle exactly what you're saying here. Is we've developed as a community 5 questions that we call the biosignature life detection assessment framework. And we want to be able to work through each of those questions when we do have an event like what you're talking about. And actually mitochondria is pretty far down on our list. Our list starts with things like, is this an authentic signal? Is this machine noise, basically? Is this really what we think it is? Is this contamination, basically? And then we ask for deeper questions. Does it make sense in this environment? Are there abiotic sources? Do I have extra information that can also confirm this? So actually getting to the mitochondria would be a few steps down on that. And what makes this a little bit different way of approaching something like, oh my gosh, I think this is an organism, is that we're really trying to bring in context clues. And what that means is this isn't just a job for Aaron or Betül or Adrian or other biologists. This is a job for the entire community, because like Betül was saying, we have to understand what is in this environment. Do we think it would even be likely here? What is its energy source? And if you gather that kind of information, you can be a little bit more certain in saying, yeah, I do think this is an organism, if you know all the things that it would take for life to be there are there as well. So this is the real nature of astrobiology, is it's a hugely cross-disciplinary thing. And we rely on our colleagues who look at inorganic and abiotic systems and the physics folks and the geologists to help us build these pictures together as well.
So if I could just join on there with Heather's answer, which was just, it's a great explanation of, where the community is at right now. So the first question we seem to ask collectively is, are you sure? And that's what we're trying to do, is collectively ask, are we sure about this? And one of the things that we've been learning a lot about, particularly recently, is the idea that chemistry can produce a lot of things that look like what we used to think of as a fossilized microorganism or something like that. So now the big question is learning how to differentiate between what's an actual ancient remnant of a microbe and what just looks like it, but that can be produced just by chemistry or just by geology. Is another way to produce it. And the problem with that is that on Earth, generally speaking, it probably is life. If it looks like life, it probably is. And now we're learning the exceptions to that. And really, it's an exception on Earth. It seems to be more exceptional that it's geology. And so when we look at life on other planets, because we have to switch our bias around, we have to assume it's geology, unless otherwise proven. It has to be extraordinary before, as a community, if we go through all the steps that Heather was talking about, before we'll all as a community go, okay, we think this may be life, but as Aaron and Betül were saying before, it could be life as we don't know it. So we have to imagine a framework that will allow us to evaluate that. And that's part of the community effort that Heather's been leading with Vicky Meadows and other people, to try to help us figure out what those standards would be if it isn't life as we know it.
So I want to assume that we found this organism and we have it on Earth and we can take it into the lab and we can study it. I think that might be one of the things you were asking about. And if we could do that, the thing that I want to know, and I sort of feel like Betül might back me up on this, because we both worked on the system a lot. The genetic system that all life uses today starts with a DNA genome that gets transcribed into messenger RNA, and then that gets translated into proteins. And that process of translating RNA into proteins is currently the oldest thing that we still have in all of our cells. And that translation process itself is not really chemically based. There's really nothing chemically similar between the stretches of RNA, what are called codons, and the amino acids, the components of the protein that they're linked to. It's really more like a naturally occurring language that allows the cell to interpret the messenger RNA and turn it into proteins. And that step seems to have opened up a whole lot of functional possibilities to life, and may have allowed life to actually unmoor itself from the setting in which it first originated. And I think you could make a case that the evolution of that translation system could be the most interesting thing that's happened in the universe. I know there are probably people at this very conference who would disagree with me, but that's what I would want to know. How does its genetic system work, and how does it bifurcate the molecules of inheritance with the molecules of molecular function? And if it does it do it in the same way that our life does it?
Yeah, I think this is great. Just to share a couple sentences there. I'm a big Trekkie, and you may recall the universal translator, right? That's I guess at the very cosmic level. And we all know, Carl Sagan said this, that we are all made of star stuff. I would want to know what part of that star stuff that life is composed of, and why. I think it would be fascinating if it is very similar to ours, and it certainly would be fascinating if it's different than ours. And what would be really fascinating is if we are wrong about everything. That would be the best outcome. That what if we are just not at all thinking about this right? And that would be the best lesson to learn from an alien.
Who would like to ask a question next?
Alex. Hi, I'm Alex Witzy with Nature. We've been talking in very interesting, very general terms, but some of us reporters are always looking for news pegs or, what's the next paper, what's the next discovery coming out? And I know it's always hard to predict when someone's going to land with a paper about phosphine on Venus or something like that. I'm just wondering what sort of specific developments and then, coming weeks to months, the rest of this year that you all are looking forward to in this space. So Adrian mentioned a experiment to go up on the space station in the relatively near future. Are there other specific developments, results of experiments, field work that you all are looking forward to in this space in the next half year to a year?
Yeah, so we've actually been working towards resurrection of ancient DNA in modern organisms to recapitulate ancestral biogeochemistry. And we've just published this in one of Cell, and on the where we resurrected Rubisco and engineered inside modern cyanobacteria to reprogram the bug. And now we're working towards applying this to inorganic chemical space. And because we actually just submitting this to where we are looking at the ancient nitrogenases, try to understand how, why these systems seem to have locked themselves down over billions of years of time scale. So that's sort of the direction that we're going. And at the same time, we're looking at the birth of translation, how this early life's machineries emerged from chemical soup. But those are a bit more computational studies that we are going towards, just to understand whether they were results of fluke accident, inevitable otherwise.
I think one of the things that we're really excited about in our group is expanding on the biogeochemical modeling work we've done to try and understand stoichiometric relationships in particles, and how we can use just those elemental abundances to diagnose if this is likely to be a particle derived from life or not. And I thought I might bring this up just in case anyone is interested in dialing in for the talk that will be on this in the 12 o'clock hour in the online session given by my colleague Chris Kempis, if you want to hear more about that. This is the kind of life detection system that could use the kind of flight hardware that has high heritage, that could fly through a plume of an ocean world, things like that, and rely on measurements that we have actually made before with this kind of equipment on other worlds.
And from my side, for the experiment that we are I'm partnering with, that is in collaboration. The PI for that project is Dr. Andreas Elster at the Free University in Berlin. There was a talk on it yesterday, and I'm going to give my talk on our participation as international conglomerate that's all working on different kinds of samples. That's going to be today at 2:30 on the online salty goodness session. And so, yeah, we're just really excited about working with all the different international partners to look at different microorganisms and comparing their biosignatures to an actual living microorganism. For us, it's high salt environments. We're looking at life that can, it's signs of life and actual living microorganisms inside salt crystals, in these little tiny droplets of water inside of a salt crystal. And we know on Earth we can find these, and we can find high salt environments elsewhere. So that's what's really exciting for us right now. And I have these amazing early career scientists that are doing incredible work that I'm just so proud of what they are developing. The creativity, the wonderful thing about extremophiles and astrobiology is that people come with their creativity. It is as much art as it is science. And so seeing their original ideas that come out of different perspectives, it's phenomenal. That's what gets me excited every day.
Thanks, all.
And if I could just back up what Betül was saying, I think this technique of molecular resurrection is really important, and we're getting better and better at it. And that I mentioned, I've talked about the last universal common ancestor of life a couple times, and that has been thought of as sort of the event horizon of biology, and you can't look past that using evolutionary biology. But it's the last universal common ancestor of organisms. It's not the last universal common ancestor of every gene. And so if we can identify the types of genes that show an evolutionary event occurring prior to the last universal common ancestor, we can begin to understand what stages of evolution looked like even earlier, and we're getting better and better at that. I don't know what everybody else in the field is doing right now, because this is my first conference in 2 years, but I know that there is the ability to do that better and to do it more is on the horizon.
I wanted to jump in here. I think I'm contractually obligated as a NASA scientist to say that there are missions that are specifically happening right now that really help address some of these questions that Betül and Adrienne and Aaron have brought up. I wanted to really point out the exciting things that are going to be coming to us from the OSIRIS-REx mission, where we'll have pristine asteroidal material that we can look at and try to understand that organic complement that hasn't, been cooked through our atmosphere, and really start to understand the diversity and complexity of our molecular inheritance from the stars. I think that really gets to Betül's question about how much of this is reflecting the universe, or how much do we differ from the rest of the universe in chemical space? And I think the other thing to talk about related to missions and Adrienne's session that she plugged, the salty goodness, is the importance of thinking about brines and what those mean for life, because brines are all over our solar system. Dragonfly will be visiting brines. There are water lenses there. The ocean worlds are very briny as well. And so understanding the relationship between life and these kinds of environments is really important too. So just be on the lookout for those kinds of things coming from missions that really actually do inform these basic science questions that we have.
Next question. I see Carl.
Yeah, I apologize for coming in a little late. I was just sort of curious to know, how do you feel? I mean, we need obviously to be looking for life as we don't know it. We need concepts to guide our search, but I'm sort of wondering about the technology. Do you feel that the technology, some of which you just been mentioning, is sufficient to tell us whether we find something really strange, something that's life, but not in our sort of our standard form of it? Or are we going to need, is there some particular technological need that we haven't yet met to be able to meet this challenge?
If I can jump in again, and I was just going to say that, one really big need is the ability to handle samples that are very salty. Those kinds of fluids are often really hard for instruments to deal with. So either new ways of interrogating those kinds of samples, or technology that can do much more sophisticated sample prep on those things is something that is a need right now. I know that's a very boring thing, desalting, but that's actually a really crucial need for interrogating these kinds of systems.
I mean, once you've sort of prepared them with all, dealing with nasty things like saltiness, I mean, if you're agnostic to, say, what's storing the genetic information or how it's being translated, then is it enough to do, some kind of mass spectrometry or some sort of measurement of different levels of compounds to say, like, this is a pattern that we think is life, or is it going to require additional technology that we just don't have?
So this isn't really a technology answer, but I would say that yes, you would always need multiple lines of evidence. I have great confidence in, the kinds of patterns that I see in chemistry, but I'm not sure if my fellow panelists would have that kind of confidence that I have. And so we have to find complementary data that can give us the confidence we would need for these kinds of questions.
I think it also depends on what sort of planetary body are you exploring, whether it is within our solar system or an exoplanet. We know that we need to get better resolution. We need to build better telescopes with higher resolution for the systems outside of ours. But when it comes to understanding our solar system, we're definitely at an advantage because things are getting better and we are able to visit these localities in situ. What is missing, however, is that it just sort of goes back to our own planet, that when we don't understand our own life and what's going on here, it's very difficult to interpret what is happening elsewhere. This also determines our priors and what we're looking for, what we think is important. It all comes from what we think we understand here. So for instance, the elemental composition or the metal composition of the planets themselves, we are not yet, there's no smoking gun yet when it comes to detecting life. I cannot tell you that if you show me this element is molybdenum and iron and this concentration and a little bit of other salts, that this planet is habitable. We are not there yet. We want to get there. And that will only be possible if we understand the laws of life, the rules of life themselves here. Because from physics and from chemistry, we understand the vastness of the space and chemical space and the universe of possibilities. And then we have a single output here. And each of these, whatever output that we recognize on our own planet is a universe of possibility in itself. But unless we reduce our understanding down to what enabled and what enables life to select from its own environment, we will not be able to interpret what we are seeing in high confidence. So these are hand in hand, technology and the scientific exploration. You cannot separate one from another.
And just to kind of pile on there with Betül saying, when we look in life in extreme environments, we are often confronted with it's too much salt or it's too acid, it'll corrode the instruments. These are questions that come up, and often it's the environments where we can't detect life that become the impetus for developing new technologies, going, well, are we sure there's no life? Because the absence of detecting life doesn't mean there isn't life. It just means we haven't detected it yet. So that's what drives a lot of technology to go, well, is it just because the environment's too extreme? Can we make a different instrument? Can we use a different sensitivity? And right now in our field, we're having an explosion of new technologies that are coming out that are enabling us to have smaller and smaller quantities of material, down to a lot of single cell detection and single cell analyses. And that's really spurring on a revolution and a renaissance and biology and life detection. So I think that translates also for looking for life, is that when we reimagine life in a different context, we have to reimagine a different instrument and to try to actually to be able to detect that. So as Betül was saying, that goes hand in hand.
I also want to add that looking for evidence for life is as hard as looking for the lack thereof, right? These are equally hard problems. What sort of information do we need to find, detect, or analyze in situ to know that absolutely no life can ever emerge here? That itself is also a very, very difficult problem. But unless, again, we understand the origins of what happened here, or rather, I think that will guide a lot of our understanding. And this is also what astrobiology is trying to do. And what's really important, I think, in the way we restructured, the way we are communicating with our peer scientists, our peers, is that we are now collected under what we call as research coordination networks. So for the first time, the astrobiology program is putting together chemists and engineers and biologists and physicists with astronomers, with scientists who are thinking about studying exoplanets. Never before we were able to create such conversations as deeply. And that includes the latest RCN that is dedicated to understanding from first cells to multicellularity. So we will see it's an astronomer and a biologist and a chemist walks into a bar, and then the engineer comes in and builds the best technology for us to make use of our knowledge. That's what we're looking for and aiming for here.
If I can piggyback off of what Betül said about understanding the origin of life, this is now a fourth reason why I think understanding early evolutionary history is important for life detection, which is that we have these prebiotic chemists who are coming up with incredible ways of making bioavailable polymers, or at least molecules that look like the kinds of molecules that biology uses, but doing it under geochemical environments. If you can make those things abiotically, that's no longer really a biosignature. And so understanding the difference between what happened before the origin of life and what happened after the origin of life can tell you whether or not you're looking at something that's a bona fide organism, or looking at something that could lead to the emergence of life, but isn't necessarily evidence of life per se.
Yeah, I wanted to follow on that and say that this is going to sound, again, boring, but the more we can understand about systems where there isn't life really helps us understand how to look for it. It's very hard to design systems for really sensitive life detection because we're living, as everyone has said, on a planet that's absolutely saturated. And the other thing about this is it's very hard for us to understand how geology and physics and planets just operate without life in the equation. And so looking at planets and that don't have life is really informative for that as well. And that becomes a way for us to understand those more subtle clues about life.
Yes, and I think what's really exciting for astrobiologists, and I must tell you that I've never seen this much enthusiasm from students, from public who are coming to universities to study, to learn about life. And I think the next generation is well aware that just because you are majoring in biology, that doesn't keep you away from learning about astronomy in a real way. It's not just a sauce, but it's truly integrated. And we know that NASA also included this as one of their top 10 priorities, is search for life. So we were very excited about that. And there are really valuable contributions in the decadal survey too. You can read in the submissions how many biologists and chemists and biochemists are rooting and are cheering for the fundamental science research to be partnered with technology for the future assessments. So we definitely would want these to be supported. I can tell you, working at a public university, that is what the public is also interested in, and that we have an amazing generation coming in who are not afraid of technological limitations, who are eager to help and work with the engineers better to find life elsewhere. It's pretty exciting. It's a good time to be an astrobiologist.
Okay, we're coming up on the time when I told my panelists that they would be free to go. I don't want to kill the conversation though. Are there some last questions from reporters that you'd like to get in?
I have just one, if you don't mind. It's been really interesting to hear sort of all the sort of theory and the ideas behind the work you're pursuing. I just wonder if you might each give me one example of a thing or a marker or a signal that you might look for based around the kinds of work that you're saying. And it might be that you don't make those sorts of predictions, but I guess that's what fundamentally people might understand. You know, Heather, for example, what kind of pattern might indicate life that isn't necessarily looking for certain molecules or something? And I just wonder if we make it real for us a little bit.
So that's an interesting question. You know, back to chemistry. Traditionally, we thought of distributions of molecules as being a way to look for life. That's something that's, you know, a deep heritage way of thinking about organic geochemistry is, you know, certain molecules arise in certain patterns as part of biological processes. And in more recent times, when we're thinking about biosignatures, chirality has been a really important thing for people to think about, is that life has this great tendency towards using molecules that have the same stereochemistry in the same system. Interestingly, those are molecules that we find out in space a lot. And then sometimes they also have chiral preferences. So that gets a little bit hard. Like, is this actually a sign of life? I think something that's coming up that I think could be really exciting and new to think about in this context of the combination of intrinsic complexity, distributions, and things like stereochemistry would be thinking about position-specific isotopes. So the heavier isotopes of the elements that we use in life are usually harder for life to incorporate into those molecular structures, but there's pattern to that. It's not just about the bulk signature of those isotopes, it's about where it's being placed in individual molecules. And I think that is going to be something that's becoming a really powerful technique as we're getting better at doing it.
Actually, I can, let me share a quick slide here just to sort of answer that question. So we think, obviously microbiologists, astrobiologists know that when you look at the sort of environment, there is life there, even though there's not maybe a visible creature. We know that there are microorganisms that give rise to this color difference. And if you're not trained as a biologist, you may think that this is empty because of the way we think what life is. What we are trying to do is to understand how these chemical mixtures turn into mixtures that have biological properties. And that's the $1 million question here. We can make molecules, we can take pictures of molecules, we can make them interact with each other, we can encapsulate them, we can put them in protocells, but yet we still don't have the experimental system that shows that this organized chemical structure can transition into a lifelike property. We have not done yet. The moment we do it, then that will be the cover of New York Times probably. So we are looking at a structure like this. So that will be an easy thing to interpret, or easier, if this was a sort of image that came from another planet. And this goes to Carl's question as well. What sort of technology are you looking for? But this is a very unlikely photo or image. Obviously the resolution will be a lot worse than this. It may just be an orange blob. What will, if we detect, or if we have some sophisticated microbes, this microscopes, maybe this will be what we will be greeting. So now let me ask you this. Do you think this is life? What do you think that this is a living organism? If imagine that this is the data that we get, we grab our coffee and morning, open our computers, and this is what something we send elsewhere is sending us. Would you think that this is a living organism? And we may debate for decades as humanity whether this is life or not. And I can tell you that this is not life. This is actually a liposome, a bunch of chemicals that are organizing themselves in the form of their chemistry. They're forming protocells. So this is a lifelike behavior in non-living chemistry. So the problem is really difficult when chemistry does these things at least at the superficial to trick us into a living behavior. What we need is to drill down. So the image alone is not going to allow us to detect life. This is why also goes back to the entire sort of the standards of evidence when it comes to detecting life. What do we need? What kind of evidence do we need? It's not going to be a single probe. It's not going to be a single image. This is an all hands on deck problem. This is going to take us decades. And I can tell you, it's going to be fun. And we will need astrobiologists trained in every aspect that are interconnected to interpret data like this to make sense of what we will be observing in decades to come.
Well, for me, the things I look at life, not at the origin of life, but life that is further along in its evolutionary process. So for me, one of the big things is the container. Life evolves inside a container. The life that we know of has a container. So we're looking at the outside, the surfaces, and where they interact with environments, and particularly where planets and life co-evolve. So we have things like iron sulfur clusters. You can either have it as a mineral, as pyrite, and that has nothing to do with biology, or it can be accumulated inside a cell, and suddenly now it's carrying oxygen and shuffling electrons around. So seeing where geology and a planet becomes encapsulated into a container and used and harnessed in a new way, those are the kind of the biosignatures that I get excited about, is this coevolution of life and the planet.
I want to sort of reiterate something that Betül said that I think is really important, which is that that we have, I don't know why, maybe we've inherited it. This is also true of the origin of life, that we have the sense that we're one great idea or one great discovery away from solving it. And we don't treat other fields this way. We don't treat medicine that way. Like we're just, you know, one idea away from living forever. It's an accumulation of discoveries over time until we build a comprehensive model of either how life originated on Earth or whether it exists on this planet or another. And I think that the search, what I see, and I'm talking as an outsider because I don't do life detection at all, but where I see the field going is this sophisticated understanding of how many different types of evidence can we pile on top of each other to make a good case that there's life on this planet, rather than we just need this and then we know that there's life there.
Last call for questions.
And I must tell Alok, you have the best questions. These are hard questions to answer. So I hope you're satisfied with our answers.
And it's been a lot of fun too.
All right, I think then I'm going to bring this session to a close. Big thanks to our panelists. You were all fantastic, and to the journalists for these great questions, as Betül told us. And we hope to see you again. We have a second session this afternoon. It will be at 1 p.m. US Eastern Time. If you can't make it, it will be recorded. See you again soon. Thank you all for coming.
Thanks for having me.